Welcome everyone. In this chapter, we are going to study enzymes. Enzymes are very important in biochemistry because they control almost every chemical reaction happen inside the body. Without enzymes, most reaction would be too slow and life processes cannot continue. Let us learn what enzymes are, how they are classified, and how they work and why they are important in pharmacy. Before we start, let us see the topic in the chapter. We will learn the definition of enzymes, their properties and their classification by the IUB system and also by molecular biology use. Then we will study the factors that affect enzyme activity, like enzyme concentration, substrate concentration, temperature, and pH. After that, we will learn the mechanism of enzyme action and common enzyme models. Next, we will study enzyme inhibitors. Finally, we will learn the therapeutic and pharmaceutical importance of enzymes in medicine and treatment. Enzymes are a specific type of protein that catalyze, meaning they increase the rate of the chemical reaction in living cell. One special characteristic of living cell is that they can perform complex reaction very quickly at a normal temperature, and enzymes are the main reasons therefore this enzyme participate an important transformation inside the cell, such as digestion, energy production, and the synthesis of biomolecules. If enzymes were not present, this reaction would still happen, but extremely slowly, so that body would not survive. Also, enzymes are usefully specific in action. And one enzyme work on one specific subtract or one specific type of reaction. Enzymes are also called biocatalysts because they catalyze reaction inside living organisms. Most enzymes are protein, but so RNA molecule can also act like enzyme. These are called ribozymes. Enzymes are produced by living cells and so the same general property of protein. Most enzymes end with the substrate AC, usually named the base and the substrate. For example, lactose is broken down by lactate. Its name is styling help in identifying enzyme easily. Now let us understand enzymes properties in the simple way. Since enzymes are protein, they show protein-like properties. They have specific isoelectrical point where they become dissolved. Enzymes are the denaturation which pH or temperature change too much. Most enzymes catalyze reaction happen below 100 degrees Celsius near normal pressure and in a neutral pH which match body condition during the reaction. The enzyme may change shape slightly, but otherwise when it return to original form and can be reused. Enzymes have monomer catalytic power. They can shape the reaction from about 1 million to 1 trillion times compared to uncatalytic reaction. IUB classification system. The IUB system classifies enzymes into six major classes based on the type of reaction they catalyze. The six classes are acetoreductase, transferase, hydrolysis, lyase, isomers, and linkages. The classification is very important for example. So remember the idea classification depends on the reaction type, not on the enzyme name. The first class in acetoreductase, the enzymes catalyze oxidation and reduction. The reaction between two substrates. One substrate is oxidized and the other substrate is reduced. In simple words, acetoreductase help in electron transfer reaction which are very common with energy metabolism and respiration. This slide continues acetoreductase. Acetoreductase includes enzymes like dihydrogenase and oxidase and reductases. They often use coenzymes like NAT or FAT. These enzymes are essential in pathways like glycolysis, TCA cycle, and electron transport chain where energy is produced. Now we move to the second class, transferase. Transferases uh, transfer a functional group from one molecule to another. Common groups transferred include phosphate group, methyl group, amino group, and glycosyl group. Transferases reactions are very common in metabolism, especially in activation to conversion step. This slide gives example for transferases. A very common example is kinase. Which transfers phosphate from ATP to another molecule. Another example is transformation, which transfers an amino group. So transferases help move group between molecule and help create a new molecule in metabolism. Now we move to the third class, hydrolysis. Hydrolysis break bond by using water. These enzymes are very important to digestion and breakdown reaction. For example, when we digested food, enzyme break carbohydrates, protein, and fats mainly through hydrolysis reaction. This slide gives example for hydrolysis. The hydrolysis includes enzymes like amylase for starch, lipids for fat, and proteases for protein. They also include phosphatases and esterases. These enzymes are found in the digestive system and also inside cell. Now we move to the fourth class, layers. The layers break down bond without using water or oxidation. They often remove a group and create a double bond, or they will add a group to double bond. These reactions are common in some metabolic pathways. Example of liases include carboxylates, which remove carbon dioxide and alcohol. Type enzyme which glide molecular pathway like glycolysis. Liases are important because they help form or break molecule without hydrolysis. Now we move to the fifth class is isomerism. Isomerism convert to one isomer into another. This means they rearrange atoms within the same molecule. This reaction to not add or remove atoms. They only rearrange the structure to that the molecule become a different form. Example for isomerism include chemomesis, epipermesis, and mutasis. These enzymes are important as carbohydrate metabolism where one sugar form is converted into another useful form. They help maintain smooth flow of metabolic pathway. Now we move to the sixth class ligases. Ligases join two molecules together to join usually requires energy commonly from A to B. Ligases from no bond like carbon nitrogen, carbon oxygen, and carbon sulfur bond. Ligases are important in the synthesis reaction. Now we see enzymes in molecular biological. In molecular biological, enzymes are widely used in laboratories and biotechnology. For example, DNA polymers is used to DNA replication enzyme, but DNA and ligase down to DNA fragment. So enzymes are not only important inside the body; they are also very important tools in the biotechnology and research. Now we start the factor affecting enzyme activity. The enzyme activity is not always the same. It changes depending on condition. The important factor include enzyme concentration, subject concentration, temperature, pH, inhibitor, and availability to cofactors. If these conditions are favorably, enzyme activity increase. If conditions unfavorably, enzyme activity reduce. First, let us understand enzyme concentration. When enzyme concentration increase, the reaction rate and generally increase because more enzyme molecules are available to bind substrate. But this increase can continue only when enough substrate in present in substrate is limited. Increased enzyme will not increase the reaction much further. Now we understand substrate concentration. With substrate concentration, will increase enzyme activity increase at first because more substrate molecule available to bind to its enzyme active side. But after a certain point, all enzyme activity side they become and enzyme become saturated. Then the reaction is a maximum speed called Vmax. After saturation, more subtract cannot increase the rate because the enzyme already working at full capacity. Now we study the effects of temperature and pH. Enzymes have optimum temperature and optimum pH. At optimum condition, enzyme activity is highest. In human, many enzymes work best around 37 degrees Celsius. If temperature increases too much, enzyme get degeneration and loss activity. Similarly, each enzyme has an optimal pH. For example, peptide work best in acidic pH, will trypsin work best in alkaline pH. If pH goes far from optimum, enzyme shape change and activity decrease. Now we learn enzyme work step by step. First, the subtract approach the enzyme and bind at the specific region called the active site. This forms an enzyme subtract complex. Then the enzyme helps the reaction to occur to lowering activation energy. After the reaction, products are formed and released from the activist side. Finally, the enzyme becomes free again and it is ready to bind to new substrate molecule. The slide continues the mechanism idea. The active side of the enzyme, the designate the perfect substrate and hold it in the correct position. This helps in the reaction occur faster. The important point in enzyme speed up reaction by lowering activation energy, but they do not change the final equilibrium. They only help the reaction reach equilibrium faster. The slide reinforces the same idea with smooth understanding. Enzymes are not smooth during the reaction. The signal enzyme molecule can act again too many subtract molecule. That is why even a small amount of enzyme can catalyze a large number of reactions in the body. Now we study two models that explain enzyme action. The first is the lock and key model. In this model, the enzyme active side like a lock and the subtract is like a key. The key is with exactly into the lock. The second is the inductive fit model. In this model, the active side flexibly and changed in the shape slide when subtract bind. So enzyme fit the subtract more perfectly. This inductive model explains the enzyme specificity more realistically. Now we study enzyme inhibitor. Enzyme inhibitor are substances that reduce or stop enzyme activity. Inhibitors are important in pharmacy because many drugs for work in by inhibiting specific enzyme. Inhibitor can be reversibly or irreversibly and they cannot different way. We and reversibly the competitive inhibitor complete within a substrate for the enzyme active side. If the inhibitor forbids the active side, substrate cannot bind and the reaction slow down. Competitive inhibitor can be reduced by increased substrate concentration. because the more subtract can out complete in the inhibitor for active state. Now we land uncompetitive inhibitor bind to the different side of the enzyme, not the active side. This change the enzyme shape and reduce activity even if subtract and present. Non-competitive inhibitor permanently inactivate the enzyme, often by forming strong bond. Instead of process increasing substrate cannot do so enzyme activity easily. This type of inhibition is important in toxicology and poisoning. Now we study why enzymes are important in medicine and pharmacy. Enzymes are used as drug target diagnosis market and sometimes as medicine symbols. Many diseases occur when enzyme activity becomes abnormal. Also, many medicine are designed to inhibit enzyme to control disease processes. So, enzymes are central in treatment and drug development. Now we look at the disease example. Enzyme involved in metabolic pathway can be targeted to treat metabolic disorder like diabetic and hyperlipidemia. In cancer also, many enzymes help cancer cells grow and multiple. If we block those enzymes, cancer growth can reduce. For example, cancer have high activity of certain enzymes and drugs are developed to target them. Now we see enzymes parasitic and antibiotic. The pest symptom enzyme are essential for survival. So parasitic can target those enzymes to kill pests. For example, acetylcholine esterase is a common enzyme target. Similarly, in bacteria, enzymes are essential for bacterial growth survival. So antibiotics often work by blocking bacterial enzyme. That is why enzyme inhibition is effective strictly in both parasiticides and antibiotic development. Finally, learn about enzyme replacement therapy. Some diseases occur because the body lacks a particular enzyme. In such cases, the missing enzyme can be given as treatment. Example include cathode disease, Fabry disease, and pump disease. So overall, enzymes are important target for therapy because they control many biological processes and controlling enzymes can control disease. This completed chapter 6 on enzymes.